CAAR T-cell therapy, a form of immunotherapy also known as adoptive cell transfer, is a highly intricate and specialized treatment. In this procedure, an expert harvests and modifies a patient's T cells, introducing a slight alteration to them. Following a period of a few weeks, the modified T cells are reintroduced into the bloodstream through a drip. Subsequently, these CAAR T cells are designed to identify and combat cancer cells. While it is a potential treatment for certain cases of leukemia in children and lymphoma in adults, individuals with other cancer types may undergo it within the framework of a clinical trial. T cells. To gain a better grasp of CAAR T-cell therapy, it's beneficial to comprehend the role of T cells. Lymphocytes, a type of white blood cells, are crucial in the body's defense against infections and diseases, including cancer. Among these lymphocytes, T cells play a significant role. T cells are mobile within the body, actively seeking out and eliminating defective cells. When the body encounters a new infection or disease, it generates specific T cells tailored to combat that particular threat. Additionally, a reserve of these T cells is retained, enabling the body to recognize and swiftly attack the infection upon subsequent exposures. What is CAAR T-cell therapy? T cells are effective in combating infections, yet distinguishing between cancer cells and normal cells poses a challenge for them. Cancer cells can evade detection by T cells, remaining unnoticed. Scientists are actively exploring methods to enhance T cells' ability to recognize cancer cells. CAAR T-cell therapy is a potential approach under consideration for achieving this goal. How does it work? Your medical team performs a procedure called apheresis to extract T cells from your blood. Subsequently, these T cells undergo genetic engineering in the laboratory, transforming them into CAAR T cells, chimeric antigen receptor T cells. These modified cells are specifically designed to identify and target a particular protein present on cancer cells. The CAAR T cells are then cultivated and allowed to multiply in the lab. Once a sufficient quantity is attained, they are reintroduced into your bloodstream through a drip. The objective is for these CAAR T cells to recognize and mount an attack against the cancer cells. The alterations made in the lab enhance their ability to persist in your body for extended periods, continuously identifying and attacking the targeted cancer cells. Ongoing research aims to determine the duration these engineered cells remain effective within the body. Who can have CAAR T-cell therapy? It seems like there might be a typo or a misunderstanding in your input. It looks like parpades is not a recognized term or part of the context related to CAAR T-cell therapy, leukemia, lymphoma, NICE decisions, or stem cell decisions. If you have a specific question or if there's a specific term or topic you'd like more information about, please provide clarification, and I'll do my best to assist you. Children and young people. CAAR T-cell therapy is recommended for individuals up to the age of 25 who are diagnosed with B-cell acute lymphoblastic leukemia, B-cell, under the following circumstances. Children or young individuals who are newly diagnosed and haven't responded to 2 cycles of treatment. Those whose leukemia has relapsed after a stem cell or bone marrow transplant. Individuals who have experienced 2 or more relapses of their disease. Children and young people whose leukemia initially responded to treatment but has relapsed and traditional chemotherapy is no longer effective. Cases where the disease has relapsed once, but a stem cell transplant is not feasible due to either the individual's health condition or the unavailability of a suitable donor. Adults. This treatment will also be accessible to certain adults diagnosed with one of the specified lymphoma types. Diffuse large B-cell lymphoma, primary mediastinal B-cell lymphoma, mantle cell lymphoma. This treatment is designed for adults diagnosed with either diffuse B-cell lymphoma or primary mediastinal lymphoma that has persisted in its growth or has relapsed after undergoing at least 2 treatments. Additionally, it is intended for individuals with mantle cell lymphoma that has continued to grow or relapsed despite receiving treatment with a specific type of targeted drug known as a Bruton's tyrosine kinase inhibitor, such as ibrutinib. Having CAAR T-cell therapy. Collecting your T cells. Initially, a tube or cannula is inserted into a vein in each arm. If the veins in the arms are inadequate, a specialized tube called a vascath may be used. One of the tubes extracts blood and directs it to an apheresis machine, which separates the various components of the blood. In the case of CAAR T-cell therapy, the machine isolates the T cells, while the remaining blood cells and normal fluid are returned to the body through the other arms too. The collection of T cells typically takes 4 to 5 hours. Subsequently, the T cells are sent to a laboratory for the production of CAR-T cells, a process that spans several weeks. Once a sufficient quantity of CAR-T cells is obtained, they are frozen for later use. Chemotherapy. Chemotherapy is administered prior to CAR-T cell therapy in a process known as lymphodepletion. This involves reducing the number of T cells in the body to prepare it for the subsequent CAR-T cell infusion. The chemotherapy is typically given over a span of several days preceding the CAR-T cell treatment. Having the CAR-T cells. The nurse with specialized training thaws the CAR-T cells in the hospital ward. Medications are administered through your vein to prevent allergic reactions. Subsequently, the cells are reintroduced into your bloodstream via a drip, typically completing in under half an hour. Throughout and following the procedure, medical professionals closely observe and monitor your condition. Staying in hospital. Following CAR-T cell therapy, close monitoring by medical professionals is essential for a duration of 2 weeks. 2 accommodation options are available, either remaining in the hospital or staying at a nearby hotel or residence, with the hospital covering the expenses. Daily visits to the ambulatory clinic, also known as AmbiCare, are required during this period, and it's worth noting that this service is not universally offered at all hospitals. Allergic reaction. It is possible to experience an allergic reaction to CAR-T cell therapy, with symptoms such as fever, chills, nausea, and breathing difficulties. To mitigate this risk, your nurse will administer medications before the treatment to minimize the chance of a reaction. If you notice any signs of an allergic response, inform your nurse immediately. Close monitoring will be conducted by the medical team throughout and after the treatment to ensure your safety. Cytokine release syndrome. Cytokines are a group of proteins that play a crucial role in enhancing the immune system. CAR-T cell therapy, designed to stimulate the immune system, can lead to a significant release of cytokines, resulting in a condition known as cytokine release syndrome (CRS). This syndrome is characterized by various symptoms, including a high fever, dizziness due to low blood pressure, and difficulty breathing. These symptoms typically manifest within the initial weeks following treatment. Side effects affecting the brain, neurological side effects. Medical professionals closely monitor patients undergoing CAR-T cell therapy for any signs of neurotoxicity. Depending on the severity and nature of the symptoms, the changes may resolve on their own or intervention may be necessary. Any neurotoxicity associated with CAR-T cell therapy. And that's a wrap for today's video. I hope you enjoyed it as much as I did creating it. If you found value or had fun, don't forget to give it a big thumbs up.